Hello, we are going to start the next session of viral immunity. I'm María José Buzón from the Vall d'Hebron Research Institute, and I'm going to co-chair this session together with Angulo from the University of Barcelona. And we are going to start with the first presentation that is called Discovery of Virally-Encoded PD-L1 Molecule by Francesc Poblador from the University of Barcelona.
Thank you for the introduction. Good evening. Today I'm going to present a part of my thesis work which is related to the virally encoded PD-L1 homologue. Viruses have evolved several mechanisms to circumvent host immune surveillance. In particular, large DNA viruses express multiple proteins known as immunoevasins that interfere at different stages of the innate and adaptive immune response. Some of these immunoevasins can come from cellular hosts, whose genes were captured during coevolution through horizontal gene transfer and incorporated into their genomes. Once there, they evolved to encode for viral proteins that either perform similar functions to those of the original cellular protein or acquire new ones. Some examples of virally encoded homologues are MHC molecules, Fc receptors, cytokines, or cell surface ligands, such as the one shown in the image, CD200, that leads to inhibition of myeloid cell activation. A vast amount of encompassed genes in large DNA viruses still remains unexplored, holding potential to encode for novel immunoevasins still undiscovered. Co-signaling molecules are proteins located in the cell surface of APCs that are crucial for T-cell activation. These proteins interact with the receptors on the surface of T cells and trigger a combination of signals that can be positive, which is called co-stimulation, or negative, co-inhibition, that give the final T-cell response when those signals are summed. Despite their relevance, it is to note that very few viral homologues of co-signaling molecules have been described up to date. Today I will focus on the PD-1, PD-L1, PD-L2 axis that triggers co-inhibitory signals during immune responses against pathogens and cancer. PD-1 is a receptor expressed on the surface of activated T cells formed by an extracellular Ig domain and that contains a cytoplasmic tail with two tyrosine motifs. Its two ligands, PD-L1 and PD-L2, are expressed inducively during inflammation, with PD-L1 being more widely expressed. Both are formed by two extracellular Ig domains and a transmembrane region, and interact with PD-1 through their N-terminal Ig domain. Upon interaction, PD-1 delivers negative regulatory signals that lead to inhibition of immune responses, peripheral tolerance, and T-cell exhaustion. However, it can also lead to tumor immune evasion. In fact, the relevance of this axis in oncogenic processes has made it a potential target for several cancer treatments. Given the importance of this axis, we hypothesize that some viruses might have captured PD-L1 or PD-L2 genes during coevolution, incorporated them into their genomes, and used them as part of their immune evasion strategies. With this hypothesis, we sought to identify viral PD-L1 or PD-L2 homologues encoded by genomes of large DNA viruses. We used the sequences of human PD-L1 and PD-L2 and performed blast analysis questioning sets of genomes of adenoviruses, herpes viruses, or pox viruses available in the gene bank. With this method, we identified DE2, a PD-L1 homologue encoded by a gamma herpes virus that infects the common bottlenose dolphin. DE2 shows a 60% of homology with human PD-L1 with an 88% of coverage and is located at the edge of the genome, which further supports its potential role as an immunoevasin. The estimated moment of capture of DE2 was calculated with a phylogenetic tree with the sequences of the Ig domains of DE2 and 84 mammal PD-L1s. In this part of the whole tree, we can see that DE2 was predicted to have been captured around 50 millions of years ago in the cetacean root. Then we characterized the structure of this viral protein. DE2 was predicted to be very similar to PD-L1, a type 1 transmembrane protein with two extracellular Ig domains that also conserves the 6 Engel glycosylation sites predicted for PD-L1. However, the viral protein does not possess a cytoplasmic tail. The overall homology between DE2 and host PD-L1 was 70%. Importantly, the identity on the IgV domain, the one related to interaction with PD-1, was 94%. We also performed three-dimensional modeling of the Ig domains of both DE2 and host PD-L1 and located some differences between them. For example, the operation of a new alpha helix on the IgV domain of DE2 and some differences in a few loops of the ICC domain. To characterize the viral protein, we cloned the gene of V2 with an HA tag and expressed it in COS7 cells by transfection. The cell lysates of these transfected cells, analyzed by Western blot, revealed a band of 50 kDa that converted into a faster migrating 32 kDa band after treatment with N-glycosidase, which supports the highly glycosylated nature of the viral protein. We also use these COS7 transfected cells to assess the location of the viral protein by flow cytometry or by indirect immunofluorescence assays using an anti-HA antibody. We located the expression of V2 at the cell surface. We then investigated the capacity of V2 to interact with the PD-1 receptor. To this end, we generated a soluble fusion protein formed by the Ig domain of PD-1 and the Fc region of the human IgG, and analyzed this interaction with COS7 cells expressing either D2 or host PD-L1 with a GFP tag. By flow cytometry, we observed that the viral protein D2 efficiently interacts with the receptor PD-1 in a similar fashion to that observed by the host PD-L1. The affinity for these interactions was calculated by surface plasmon resonance analysis, testing the binding of the PD-1 to increasing concentrations of D2 or host PD-L1. As you can see here, the viral protein shows a high affinity and a stable interaction with the host receptor, again in a similar fashion to PD-L1. I'm not going to enter into much details, but performing similar types of analysis, we also observed that D2 does not interact with human PD-1. Then we asked whether D2 could impair T-cell signaling via PD-1 engagement. To this end, we use a fluorescence-based cellular approach designed to investigate human T-cell co-signaling pathways. First of all, since D2 does not interact with human PD-1, we constructed a chimeric molecule formed by the Ig domain of host PD-1 and the transmembrane region and cytoplasmic tail of the human PD-1. Then we generated a Jurkat reporter GFP cell line, a T-cell line, that expressed this chimeric molecule in its cell surface, and several T-cell stimulator lines, or TCS, that express either D2 or host PD-L1, or none of them, at its cell surface. These TCS also express an anti-CD3 single-chain antibody that engage on the CD3 TCR complex of the Jurkat reporter cells, and this gave a reported NF-kappa beta reporter activity. As you can see here, the engagement of D2 efficiently decreased T-cell activation in a similar fashion to that observed by host PD-L1. Moreover, when we carried out these assays in the presence of the co-stimulatory molecule CD86 that engage on the CD28 of the Jurkat cells and strongly enhanced the reporter activity, the T-cell co-stimulation was strongly reduced in the presence of D2, in this case in a higher degree than the host PD-L1. Finally, we wanted to assess whether D2 retains the ability to interact with CD80 in FIS. This interaction has been reported to prevent the PD-1-PD-L1 interaction, therefore blocking negative signaling, while allowing CD80 to interact with CD28 and give co-stimulatory signals, which will be negative for viral immune evasion. To this end, we co-transfected COS7 cells with either D2 or host PD-L1 and with CD80 with an HA tag, and analyzed its interaction with host PD-1. The flow cytometry assay showed that the engagement of PD-L1 with PD-1 was strongly reduced in presence of CD80, but that was not the case with D2 and PD-1, which was largely unaffected. To assess the origin of this difference, we constructed several mutants with changes on the IgV domain of PD-L1 by amino acids of D2. One of these mutants, the one that contained the mutations responsible for the apparition of the alpha helix I mentioned before, showed a drastic reduction on the percentage of blockade by CD80. This means that these two amino acids, and therefore the apparition of this new alpha helix, are the responsible for the loss of D2 and desired ability to interact with CD80 in FIS. As a final conclusion, D2 represents the first functional viral PD-L1 homologue identified and points to a new viral immune evasion strategy. This work has been done in the cytomegalovirus group led by Dr. Angulo at the University of Barcelona, with contributions of former lab members Pablo Martínez and Domènec Farré, and in collaboration with Dr. Pablo Engel of the same Immunology unit and Drs. Peter Steinberger and Judith Leitner of the Medical University of Vienna. Thank you for your attention.
Do we have questions? I'm going to start with the first one. Have you tried to use the drugs that are used in the clinic to inhibit the axis PD-1, PD-L1?
The what?
The drugs that are used in the clinic for patients to inhibit the interaction?
We haven't tried any drugs for this treatment.
Interesting. Any other question?
Do you know which viruses are have incorporated these PD-L1 similars?
This virus or other viruses?
Other virus, yeah.
We still don't know yet. I mean, the number of viruses that are encoded in the gene bank is limited. In this search, we located this one. Maybe there are other homologs that haven't been discovered because the percentage of homology is too low, or maybe there are other viruses that still haven't been, still are not in the database and maybe expressed.
I missed something. It's just, you probably have explained it, but I just missed it. Yeah, you have said that D2 doesn't interact with human PD-1?
Does not interact.
Does not. No. So which is the host of the virus that encodes for D2?
What?
So which is the host?
The host is dolphin, sorry.
Is?
Common bottlenose dolphin.
Okay.
The host.
Okay, yes, thank you.
The host for the virus.
Thank you.